Hello friends, welcome back to a new video, that is MCQs on developmental biology, and this is the part 1 of this series of videos on developmental biology. So let's start the question answer session on developmental biology. So the first question of this video, that is the Mullerian duct in the female amniotes develops into option A utera, option B oviduct, option C uterus, or option D seminal receptacle. So the right answer about the Mullerian duct is option B, that is oviduct. So the Mullerian duct in the female amniotes develops into oviduct, and this Mullerian duct is the embryonic structure that develops into the female reproductive tract, including the oviduct, uterus, cervix, and upper vagina. Okay, so these are all about the Mullerian duct, and here in this picture you can see these tubes are the Mullerian ducts, and after development they will fuse with each other and ultimately produce the uterus. Okay, so this is the development of uterus. So let's move on to question number 2, that is the term gray crescent is associated with the egg of option A branchiostoma, option B frog, option C chick, or option D frog and chick. And the right one about the gray crescent is option B, it is frog. So the term gray crescent is associated with the egg of frog. So what is gray crescent? This gray crescent is an area of the region opposite the sperm enters that formed a melanin rich place. So where the sperm enter into the egg or ovum, the opposite side of the sperm entry is known as the gray crescent. There are two poles, vegetative and animal poles, in the after fertilization. So the gray crescent, which is rich in melanin, separate these two poles, that is vegetative and animal poles. They differentiate those two poles like this. This middle portion is the gray crescent, okay, and this is the animal pole and this is the vegetative pole. Okay, so let's move on to next question, that is question number 3. So after fertilization, option A zygote divides first to form embryo, option B primary endosperm nucleus divides first to form endosperm, option C both undergo a long period of rest, option D both divide simultaneously. And the right answer, the thing occurred after fertilization, is option B. It is primary endosperm nucleus divides first to form endosperm. So after fertilization, primary endosperm nucleus divides first to form endosperm. And here endosperm is a tissue that surrounds and nourishes the embryo in the seeds of angiosperms, that are the flowering plants. Okay, so next question देखेंगे. Next is organ identity genes are responsible for correct positioning of floral organs on floral meristem. So mutation in them, that means mutation in that organ specific gene, will lead to option A loss of organ from certain holes, option B more number of organs in certain holes, or option C appearance of organ at incorrect positions, or option D no flower. So which one is right answer about the mutation on the organ identify genes? And the right one is option C, that is appearance of organs at incorrect positions. So if mutations are occurred in the organ identified genes, then appearance of organs at incorrect position will be found. Okay, so let's move on to question number 5. That is the lateral separation of amphibian embryo at two-celled stage will result in option A identical twins, option B two embryo joined at belly region, option C single embryo, or option D, two embryos missing various organs. So which one is right about the lateral separation of amphibians embryo at two-cell stage? And the right one is option A, that is identical twins. So the lateral separation of amphibian embryo at two-cell stage will result in identical twins. Okay, let's move on to question number 6, in which the first division of the zygote is option A transverse, option B vertical, option C variable, option C oblique. So the right answer is option A, that is transverse. So in wheat, the first division of the zygote is transverse division. Okay. Next is question number 7. That is milky water of green coconut is option A liquid chalaza, option B liquid nucleus, option C liquid nuclear endosperm, option D liquid female gametophyte. And the right one is option C, that is liquid nuclear endosperm. So let's move on to question number 8, that is which is necessary for seed formation, and the options are option A megaspore should not release from ovary, option B flower, option C closed ovary, option D time gap between pollination and fertilization. So which one is necessary for seed formation? And the right one is option A, that is megaspore should not release from ovary, and that is the necessary condition for seed formation. Ovary should not, sorry, megaspore should not release from ovary. Okay, and let's move on to question number 9, that is in a two-celled embryo the cell towards the antipodal end is termed as option A embryonal cell, option B suspensus cell, option C epiphysis, option D hypophysis. And the right one is option A, that is embryonal cell. So in a two-celled embryo, the cell towards the antipodal end is termed as embryonal cell. So let's move on to question number 10. It is a single large shield-shaped lateral cotyledon in monocot embryo is called option A scutellum, option B alluron, option C tegulum, or option D epiblast. And the right one is option A, that is scutellum. So a single large shield shaped lateral cotyledon in monocot embryo is called scutellum. Okay, and the scutellum is a first leaf of a monocot plant. It's also known as the first primordial leaf. It's a large shielded cotyledon that is located on the lateral side of the embryonal axis. Okay, so let's move on to question number 11. It is the name for the type of cleavage in the mammalian embryo is called option A planar cleavage, option B unequal cleavage, option C rotational cleavage, or option D radial cleavage. And the right answer about the type of cleavage in the mammalian embryo is option C, that is rotational cleavage. So the name of the type of cleavage in the mammalian embryo is called rotational cleavage. Next is question number 12, and that is during cleavage the cell division is very rapid. So the daughter cell do not undergo any growth and the cells thus become gradually smaller in volume. So hence options are option A, there is no growth in the volume of the embryo, option B, the embryo grows in volume, option C, the embryo becomes smaller in volume, or option D, the embryo remains static. So which one is correct answer? So the right one is option A, that is there is no growth in the volume of the embryo. So during cleavage the cell division is very rapid. So the daughter cells do not undergo any growth and the cells thus become gradually smaller in volume. Hence there is no growth in the volume of the embryo. Okay. So let's move on to question number 13, and that is, what does not happen during gastrulation? What does not happen in gastrulation? And the options are: option A, three embryonic germ layers are established; option B, large scale migration of cells to organize the three germ layers; option C, the embryo increases in size; option D, developmental fates of the germ layers are decided. And the right one is option C, that is the embryo increases in size. So what does not happen during gastrulation? That is the embryo doesn't increases in size. Okay, so other the things, that is three embryonic germ layers are established in gastrulation, large scale migration of cells to organize these three germ layers, and the developmental fates of the germ layers are decided in this gastrulation stage. So here we will see this gastrulation is a stage in the early development of most animals. It is the process of an embryo transforming from a single layered ball of cells called the blastula to into a multi-layered ball of cells called a gastrula. So during the development of animals, this is a process where single-celled blastula cell transformed into multi-layered gastrula cell. Here you can see this is the picture. Here this is the blastula and this is a single layer, and here you can see three-layered gastrula stage. Okay, so the outer membrane is called ectoderm, then in middle, that is the blastocells is there, and the middle is the endoderm. Okay, and this is the acurome. Okay, and this is the blastopore. So this is the gastrulation stage, and let's move on to next question, that is question number 14. And a blastula composed of densely packed but larger sized and relatively smaller number of cells without or extremely small blastocylic space in the center is called: option A, stereoblastula; option B, coeloblastula; or option C, discoblastula; or option D, all of the above. So which one is right? That is option A, that is stereoblastula is the right option. So a blastula composed of densely packed but large sized and relatively small number of cells without or extremely small blastocylic space. So there is very less. This one is the blastocell. So in which blastula cells the blastocell is very less, and that is the stereoblastula. So here you can see this is the outer side of the blastula, and this is the blastocele. This portion is called the blastocele. So in which blastula the blastocele is very less, that blastula is known as stereoblastula. So a stereoblastula is a solid blastula which is a blastula without a cavity. It is characterized by the absence of a blastocele. So stereoblastula is formed by spiral cleavage. Okay. Next is question number 15. So which of the following pairs are not correctly matched? And the options are: option A, blastocele related to blastula of insect; blastopore is related to blastula of frog; option C, blastocyst is related to blastula of man; or option D, blastodisc is related to blastula of bony fish. And which one is not correctly matched? That is option B, that is blastopore is not related to blastula of frog. Okay, here you can see the blastula of frog is hollow and has a very well developed blastocele. It is said to be a coeloblastula. Okay, so and here you can see this is the blastula of frog. So let's move on to, that is the coeloblastula. Okay, so let's move on to next question, that is question number 16. Here, Ingregation is option A, migration of individual cells from the surface layer into the interior of the embryo. Option B, ingregation is movement of epithelial sheets. Option C, inward movement of outer layer. And option D, splitting of one cellular sheet into two or more. And the right one about the ingregation is option A, that is the migration of individual cells from the surface layer into the interior of the embryo. So here you can see this whole is an embryo, and the surface cells, these are the surface cells, membrane cells. So here these cells run or move towards the embryo, and that is known as, and that migration of individual cells into the embryo is known as ingregation. This thing is happen in Drosophila. Okay, so let's move on to next question, that is question number 17. So which of the following is unique to internal fertilization? And the options are: option A, many zygotes are formed at one time. Option B, an egg changes once it is fertilized so that no more than one sperm can enter to this embryo. Or option C, every zygote develops into an embryo. Option D, sperm are deposited inside the female body. So which one is unique to internal fertilization? And the right answer is option D, that is sperm are deposited inside the female body. That is known as the internal fertilization. So here internal fertilization is a process of fertilization that occur inside the body of an individual. It occurs mostly in the land animals. For internal fertilization to occur, there needs to be a method for the introduction of male sperm into the female reproductive tract. Okay, so let's move on to question number 18, and that is, which of the following statements is or are incorrect about mammals? And the options are: option A, fertilization occurs in the ampulla of oviduct. Option B, oocytes complex meiosis before sperm entry. Oocyte complete meiosis before sperm entry. Or option C, cleavage is rotational type. And option D, cleavage shows asynchrony, that is not divided at the same time. So which one is incorrect statement about mammals? And the right one is option B, that is oocytes complete meiosis before sperm entry. That is not correct about mammals. So the next one is question number 19. So in vertebrate animals, spermatogenesis and oogenesis differ in that options are: option A, oogenesis begins at the onset of sexual maturity, whereas spermatogenesis happens in embryonic development. Option B, oogenesis produces four haploid cells, whereas spermatogenesis produces only one functional spermatozoon. Or option C, cytokinesis is unequal in oogenesis, whereas it is equal in spermatogenesis. Or option D, spermatogenesis is not completed until after fertilization occurs, but oogenesis is completed by the time a girl is born. So the right answer about the spermatogenesis and oogenesis difference is option C, that is cytokinesis is unequal in oogenesis, whereas it is equal in spermatogenesis. So here you can see this spermatogenesis and oogenesis are the process of formation of male and female gametes. Spermatogenesis leads to the formation of sperms, whereas oogenesis helps in the formation of ova or egg. The fertilization of sperm and ova leads to the formation of a zygote, which further develops into an embryo. So oogenesis is the process of producing ovum or egg, and spermatogenesis is the process of producing sperm. Okay, so let's move on to And the right answer is cytokinesis is unequal in oogenesis, whereas it is equal in spermatogenesis. So let's move on to the last question of this video. That is, morphallaxis can be defined as option A, production of lost organ by division in remaining cells, option B, reinitiation of cell division in existing cells followed by repatterning of those cells, option C, production of complete organisms by single cell, option D, movement of organism toward stimulus. And the right answer is option B, that is reinitiation of cell division in existing cells followed by repatterning of those cells. So morphallaxis can be defined as the reinitiation of cell division in existing cells followed by repatterning of those cells. So what does it mean? That is, morphallaxis is the regeneration of specific tissue in a variety of organisms due to loss or death of the existing tissue. So if a tissue is dead or lost due to some accident, then morphallaxis is the regeneration of the specific tissue. Okay, so an example of morphallaxis in lower animals is budding in hydra, and the new hydra grows by the loss of cells from the parental body. Okay, so these are all 20 questions on this part 1 video on developmental biology. Okay, so thank you for watching this video.